Welcome back. Our objective, I want to remind you, is an in-depth description of the inner workings of cancer. Hopefully, understanding will lead to better treatment. Like a car, two things are needed to make a cancer cell grow. First, oncogenes, which stimulate cell division, and second, removal of the inhibition by tumor suppressor genes. In the last lecture, we described the genes changed in cancer, proto-oncogenes and oncogenes, in a generic term, and now we want to find out exactly what they do. Proto-oncogenes are mutated in 90% of cancers that are not, and these are the cancers that are not virus-caused. And the first question we can ask is, do they act alone? Can one proto-oncogene mutation cause cancer? Well, there was an early experiment on the Src gene with Rous sarcoma virus and indicated, well, that could be, but it's not usually so. Here's an early experiment that showed that. This experiment described two oncogenes, which we will be describing later on. One is called MYC, and the other is called RAS. And mice were given these genes in an activated form, and this is a Kaplan-Meier survival curve. You see on the y-axis is percentage of tumor-free animals. We're looking for for tumors, and ultimately the tumors do kill the animal, and the time in days. If MYC was given all by itself, you can see that the curve kind of went down after about 100 days, but really didn't go down rapidly. If RAS was given separately to a group of animals, it went down somewhat, but not as much. But if the two were given together, the whole was much greater than the sum of the parts. So now we know that more than one proto-oncogene in a cell that's not virus infected has to be turned on. The question now arises, what do proto-oncogenes do in the cell to turn on cancer? And the second question is, how do they get turned on? How do the proto-oncogenes all of a sudden become active? We know the virus brings them in an active form, but they're sitting dormant in my cells, hopefully, and they're not turned on at the moment. So let's describe this. First of all, I will describe oncogene functions. There are three of them that I will describe. First, some oncogenes make products that control gene expression. Remember, that's the synthesis of protein from a sequence of DNA. And they usually do this by turning on genes involved with cell division. The second examples I'll describe involve cell signaling. Some oncogenes involve this system of a growth factor or substance outside the cell, binding to the cell and stimulating cell division that way by knocking on the door and making some action go on in the cell nucleus. Third, there are some oncogenes that control cell death by reducing it. And you'll recall the argument that a tumor grows by adding cells, but also by not having cells die. So in the last lecture, I described a couple of things about oncogenes and proto-oncogenes. First, about the level of gene expression, and then how tumor viruses have oncogenes and normal cells proto-oncogenes. Now let's see what those proto-oncogenes do. And the first aspect is going to be that some of them control gene expression. And you'll recall in a previous lecture, I described DNA and RNA and the protein synthesis system. And this begins at a place called the promoter. You remember that ignition switch right at the start of a gene that essentially controls whether the rest of the gene is going to be expressed. There is an exquisite, that's the only word I can use, mechanism of control at the promoter. I made this really simple for you in the last lecture. I said, well, you know, there's a protein that will bind to the promoter, and that'll kind of open it up or encourage just to turn on the switch to make it go, okay? Doesn't turn out that way. At most promoters, there are at least 15 proteins piling on. So here is an example of a promoter with just some of the proteins piling on. You see them there. A whole bunch of proteins are piling on to the promoter. So they're repressors and activators, and it's a sum total of all of them that determines whether something is going to happen. It's like a child being shouted at by all sorts of people saying, do this, don't do this, do this, don't do this. Finally, the kid kind of evaluates all of them and does what the majority say. I'm going to give you an example of an oncogene that acts at the promoter as a kind of paradigm for this whole system. And the oncogene I'm going to talk about is MYC, M-Y-C. There is a virus called the avian bird myeloid is a blood tumor, leukemia. And the oncogene it carries into the bird cell is a MYC oncogene, MYC. So you see where we got that number because that name because it's a myeloid leukemia. The bird DNA, the host DNA, has also a proto-oncogene called MYC. And MYC is, of course, present in many vertebrate organisms, including humans. I have MYC in my genes, and so do you. In normal cells, MYC is expressed typically during cell division, when cells are reproducing. In cancer cells, MYC expression is uncontrolled. A lot of it is being made. MYC is uncontrolled in 80% of breast cancers, 70% of colorectal cancer, 90% of gynecological cancers, 50% of liver cancer, 80% of lymphomas. Like, for example, Burkitt's lymphoma, the lymphoma of the jaw of children I described in the previous lecture, has MYC activated in 80% of the cases. A tumor I want to focus on is called neuroblastoma. Neuroblastoma is a childhood tumor of the central nervous system. Neuroblastoma is much more treatable than it was previously. But initially what people did was they found that the MYC gene was amplified. I'll come to that later on in the course about amplification of genes. But the most important thing is there was more MYC protein and it correlated with a poor prognosis. So here is a graph showing initial studies about 20 years ago of children with neuroblastoma, and they would biopsy the neuroblastoma. And the children who had a lot of MYC overexpression, a lot of this proto-oncogene being overexpressed, had much worse prognosis. Fortunately, and this is a good example of how molecular biology has helped, children who had this MYC overexpression were treated aggressively with therapy, and now they're doing much better than they did in the past. So here's where molecular biology allowed us to stratify patients and choose those patients to which we're going to give aggressive therapy. How does MYC do it? MYC is a transcription factor. MYC is a protein that acts at the promoter. In fact, if you look at the structure of MYC, you see something very similar to what I showed you in the last lecture. Remember this protein that bound to DNA, and it kind of had these arms that went out to DNA and recognized the DNA. And in fact, in the case of MYC, it has a specific sequence in the promoter it's looking for. The sequence is called CACGTG on one of the DNA strands. So the MYC has a protein structure that goes in and just looks for that specific sequence. Where is that sequence? It's in certain promoters. And what MYC does is unwind the DNA a little bit because it's sitting right inside the DNA. So it unwinds the DNA a little bit, exposes it, so other proteins can come in and do their thing. And that thing may be to block transcription or to turn on transcription. Now, MYC is usually off, right, except during cell division. But MYC goes on, has a lot more expression in cancer cells. So the result is more DNA is available for the regulation of expression. But MYC is one of these transcriptional regulators. It's binding to DNA just the same because it's allowing the other things to come in and do things. What does MYC do? It upregulates some genes. In other words, MYC binds, and then other proteins come in to upregulate, to have more expression of certain genes. What are those genes? Cell division kinases. You remember those? Those are things that allow cell division to go on. Maybe they inactivate a tumor suppressor, whatever, right? They're things that allow cell division to proceed. MYC causes the upregulation of response to growth factors. So growth factor will bind to a cell and be more active in terms of its results of its binding because MYC is sitting there in the nucleus and says, come on here, you can go ahead and stimulate cell division. On the other hand, MYC opens up promoters that attract things that turn off expression, such as cell adhesion proteins. You remember in metastasis, when cancer cells spread and have to go into the bloodstream, etc., they have to detach from one another. Well, if a cell detaches, very often it's losing its adhesion proteins, those proteins that allow it to stick. It loses those adhesion proteins. And what MYC does is lower the synthesis of adhesion proteins because it attracts proteins that do that. Another thing it does is it lowers the number of proteins involved in cell differentiation. And you recall, and I've said this several times in the course, that differentiated, specialized cells don't divide. So MYC has these effects on different promoters, all of which have the same DNA 6-nucleotide sequence. MYC lands there, opens it up, and attracts those other proteins that then either turn genes on or turn genes off. But the net effect is more cell division and cancer. So that's the example I'm going to give of a protein that's involved in the regulation of gene expression. Some cancer genes control cell signaling. Now, I mentioned in a normal differentiated cell, it's not dividing, and cancer cells are dividing. You'll recall cell reproduction is controlled in two ways: internally by these kinases, etc., and externally by growth factors or proteins, something binding to the outside of the cell, binding to a receptor protein, and then the receptor protein does something to stimulate the inside of the cell. So here is the system in slightly more detail than we saw before, in molecular detail. The signal is a growth factor that comes from somewhere else outside of the cell. It binds to a receptor. Receptor binding somehow causes a change inside the cell, and then there are signaling proteins inside the cell, like a bunch of dominoes that kind of fall, right? One pushes another. Many of these signaling proteins are protein kinases. Very interesting. So a protein kinase will get activated. I mentioned before there are hundreds of protein kinases in the cell, many involved in signaling. So one of them will get activated and kinase, or put a phosphate on its target protein, which is the next step. And the next step gets activated, and it puts phosphate on the next step, and it puts phosphate on the next step, etc. And the end result of this signaling pathway is usually a transcription factor in the cell nucleus that alters the expression of genes. So that's the external control of this cell division. Now, here's the interesting thing about this. I've presented a very simplified pathway here. But what's amazing about it is there are oncogenes at almost every step. If you have a stack of 20 dominoes, say there's 20 steps, and sometimes there are, and the objective is that domino 20 fall, and that's the transcription factor that's going to affect DNA expression. It doesn't matter whether you push down domino 1, because if you push down domino 1, all the rest of them fall, 2, 3, 4, 5, 6, right, all the way to 20. Or you can push down domino 10, you can do all of them, right? So if we have a mutation in domino 16, or step 16, such that it becomes active without being stimulated by the others, in other words, the domino falls, you can see that domino 20 is still going to fall. It turns out that many of the intermediary steps can be oncogenes, because if you turn one of them on, all the subsequent steps get turned on. Well, rather than give you this complicated thing, let's go back and zero in on one of them. And the one gene I'm going to talk about is RAS, R-A-S. It was isolated first in a rat sarcoma. Sarcoma is a muscle type of tumor. And the proto-oncogene is called RAS, and it's a protein at the cell membrane. Here's what RAS does. It's the first of the dominoes. After a growth factor binds to the receptor jiggles somebody, and sitting right there is a molecule called RAS, and then the RAS becomes active. RAS actually changes its three-dimensional structure indirectly by binding with the receptor protein inside the cell. Now, here's a question. What if the gene that codes for RAS was mutated such that the RAS protein was changed so it's always active? It doesn't need this push from the outside. Well, then in the sarcoma, which is exactly what happens when it's a non-virus infected situation, in the rat sarcoma, you have this abnormal signaling. The RAS protein is always on. You don't care about growth factor anymore. The RAS protein is always on, and it stimulates all the other dominoes in the pathway, and finally you end up with an active transcription factor in cell division. 50% of human cancers have an activated RAS. As you'll see, it's a very common mutation in human cancer. Application, let's look for something that blocks RAS. So people have tried to design drugs that block RAS. It's a kind of tough situation. Here's an example of something that blocks RAS. There is a small, mild virus called a reovirus. It's a subclinical infection. It really doesn't cause a big problem. It does infect humans. And it turns out RAS, the reovirus, doesn't usually duplicate inside cells. But if this, for good molecular reasons, if the RAS, the cell has been activated, it's got an active RAS, and the RAS is really turned on and extremely active, the cell gets selectively killed by a reovirus infection. So this is called an oncolytic virus. Onco, cancer, lytic means the cell busts. So it's an oncolytic virus that kills RAS-activated cells. This is actually now in some clinical trials for melanoma and breast and colon and prostate cancer. But RAS is a tough target because even though it is mutated in many cancers and very active, it is also active in many, many other cell types. So the side effects of just blocking RAS probably are going to be pretty severe if that's selected as a therapy. Be that as it may, if we look at all the other dominoes, all the other steps in signal transduction pathway, there are many targeted therapies that are being developed for each one of those steps because all we got to do is block one of them. All we got to do is inhibit one of the dominoes from falling, and the last domino on the list will not fall. So many of these are protein kinases, these intermediate steps. So there's a whole family of targeted drugs in therapy now called protein kinase inhibitors. The very first lecture of the course about Kareem Abdul-Jabbar, the opening story, I described Gleevec, a drug. It is an inhibitor of a protein kinase that is involved in downstream signaling. It comes in and replaces one of the dominoes toward the end of the list. And it's very active, and it causes the transcription to be activated. And so we block, with Gleevec, we can block that domino from working, and the cancer is alleviated. So that's the second type, right? First we talk about gene expression, and then I talked about oncogenes, proto-oncogenes that are involved with signaling. The third type of cancer-causing gene I'm going to talk about is controlling cell death. Okay, so viral oncogene, cell proto-oncogene types have been described for many types of tumors, and especially in animals. Here's one. It was called B-cell lymphoma. A B-cell is a white blood cell that has to produce antibodies, and lymphoma just is a tumor of it from the lymph node. So there is a gene that was characterized called BCL2. It was brought in by a virus in animals, but also as a proto-oncogene gets activated. Initially it was thought it's an oncogene, it stimulates cell division, that's what it does. However, in a certain type of cancer called follicular large cell lymphoma, it's just one of those lymphomas that comes from the line of differentiation of white blood cells, a doctor named Stanley Korsmeyer at Washington University in St. Louis showed that the BCL2 protein is rather different. It's present where cells don't die, and it's absent where they die. In other words, the BCL2 protein seems to block programmed cell death. This was a complete surprise. It was a totally new way of looking at the development of cancer. Because initially we thought oncogenes are the gas pedals, right? They make things go, and tumor suppressor genes block it. We didn't think of cell death as being involved at all. It turns out some oncogenes and proto-oncogenes, when they're activated, block cell death. And the ultimate proof of this was when mice were genetically engineered to have an overactive BCL2. Now, you can imagine, overactive BCL2 means no programmed cell death. What happens? They get tumors. White blood cell form tumors, and other cells form tumors because the white blood cells don't die. You recall, I told you earlier on in the course that white blood cells typically will last about 2 months in the bloodstream, and then they undergo programmed cell death. Now they're not going to because the BCL2 is active. Then they created some animals that have no BCL2 gene. The gene was knocked out. What's going to happen there? Cells are going to go wild, essentially. And it turns out that during development, during the embryo formation and fetal formation, Programmed cell death is a very common occurrence. I mentioned to you before that when digits form, there is a tissue between the digits, and the tissue has programmed cell death. But that's the least of the problems if you don't have a BCL2 gene active. If that's not the case, there's going to be chaos in the embryo, and the animal doesn't get very, very far. So the role of BCL2 is to prevent apoptosis. How does it do it? Well, normally, if there is DNA damage, the DNA gets repaired and the cell is okay. If there is extensive DNA damage, the cell will sense that and go into programmed cell death, and that's it. There'll be execution signals and the cell dies. If BCL2 is overexpressed, however, the apoptosis doesn't happen, and what's going to happen? The cell is going to survive, and mutations will accumulate in cells that normally would have died through programmed cell death. This has been shown to occur in human melanoma, in breast cancer, in prostate cancer, in lung cancer. So how can we get around this? Of course, try to design a drug that blocks BCL2. And so a number of these are in the market. Some of them are actually out. Here's one example. Here are blood cells before treatment in a leukemia. And you can see that after treatment, the leukemia has been alleviated quite a bit. The blood cells are the blue stain cells. Leukemia has been treated with an anti-BCL2 drug. And so now they will go into programmed cell death and the cells will die. Now, I should emphasize I've given you a simplified view because there are many other proteins involved in apoptosis, and of course, drugs are being designed for all of those different pathways. So now I've given you a couple of ways that oncogenes and proto-oncogenes inside the cell work. But I haven't told you yet how they become activated in cancer. I've told you in the last lecture they can be brought in active form by a virus, but remember, 90% of the time, cancers occur because of proto-oncogenes inside the cell are somehow being changed by mutation. 3 different ways that they can be changed in cancer. Those 3 ways are to be amplified, more copies of the gene. Secondly, to be translocated. A gene could be translocated to an active promoter. I'll discuss that in more detail. And finally, they can be mutated. Actually, the gene can be mutated into an active form. And I will give you specific examples and specific tumors for all 3 of them. First, amplification of a proto-oncogene. You remember I talked earlier on about this childhood tumor, neuroblastoma. It turns out that the way that these children have more MYC protein, the MYC transcription protein in neuroblastoma, is that they have more copies of the gene. Here's a more recent clinical example. It's an example of a protein called HER2, human epidermal growth factor receptor 2. It's a receptor for a growth factor. And in 30% of breast cancers, HER2 is amplified. The gene is amplified. And you recall I described previously that more copies of a gene means more copies of its protein. So here is a normal cell, which got a couple of these receptors on it. And here in this instance of an amplified HER2, and 30% of the time, are more copies of the receptor. Obviously, if a growth factor comes, this growth factor that binds to it, there's going to be more HER2 binding to the HER2 binding, and the more HER2 binding will lead to more cell growth. It's a prognostic indicator for cancer. And now I'll give you a very historic graph, and it's historic in terms of therapy as well. This is a prognostic indicator, so the probability of survival here, it's a Kaplan-Meier curve. On the y-axis, it shows survival. On the x-axis is time after initial diagnosis. And you see here that if the HER2 gene is non-amplified, there's a certain survival curve. And if you see it's amplified, there is a much worse survival. And you'll note the date, 1990, because I put in that date for a reason, because in the 1990s, a drug was developed to essentially treat overexpressing HER2 breast cancer. The drug is called Herceptin. I'll describe later on how it works, but essentially binds to the receptor and prevents the receptor from doing its thing. And that drug has taken that curve that goes way down and brought it up to the other curve. In other words, it's really extended the lives of hundreds of thousands of women in use in treatment. I'll describe that in some more detail later on. But this is a really good example of using molecular biology as a prognostic indicator and then taking that prognosis and then targeting therapy for it. Second method of an oncogene is by activating by translocation. You remember in the first part of the course first lecture I described Kareem Abdul-Jabbar and a shuffling of a chromosome. Well, one of the types of shuffling I think I've mentioned before is to make a fusion gene. Remember the two genes were half of one gene and half of the other gene when there was a swap between two chromosomes, a fusion gene. Another type is to take a gene that is normally not very active and to put it by a very active promoter. It's kind of like taking a normal Midwestern human being and putting them into Las Vegas. Things change. They get activated. Well, this gene becomes activated in a translocation event. That's a mutation. The gene is getting translocated. The gene I'm describing here, is BCL2 in follicular lymphoma. BCL2 is normally not very active. It's on chromosome 18. And in follicular lymphoma, in this cell that's being turned on to make antibodies, it's an immune system cell, it's a white blood cell. The BCL2 gene, through mutation, gets translocated to the promoter for the antibodies. And the promoter for antibodies is really active. It's attracting all these proteins and turning on, et cetera, et cetera. So now the BCL2 gets active in getting turned on. Its protein gets made, less apoptosis and cancer. Chromosome abnormalities with translocations are very common in cancers. 50,000 different ones have been reported in the literature. There's so many of them. And they're typical on solid tumors. Typically in blood tumors, there's one translocation, but in solid tumors of solid organs, there can be many translocations. It's not clear how this happens. How does translocation happen? It seems to be most common in radiation damage, where you get a double-stranded break of DNA, but it's hard to know exactly how it happens, and it may be due to poor repair. Not clear. But it does happen, and it's one of the methods of activating proto-oncogenes. So that's the second method, the first having been the method of amplifying the gene itself. The third method is mutation. So here is RAS here in the inside of the cell, and we'll close up on RAS here. And when RAS becomes active, if RAS becomes active, okay, and RAS could become active by a mutation that changes its shape. And if it changes its shape so it's always on, then we've got a big problem because the dominoes are always going down in the cell rather than being controlled by something outside of the cell. RAS signaling is involved in colon cancer, abnormal RAS signaling in colon cancer, in lung cancer, in melanoma, in pancreatic cancer. And again, it's typically mutations in the RAS protein, single mutations in the RAS protein, such that it doesn't need a kick in the pants anymore to get going. It just gets going without regard to outside stimulation. This graph shows you it's a Kaplan-Meier curve for bile duct cancer. Overall survival is very poor if there is a RAS mutation. Here's another cancer in which there's a RAS mutation. Skin cancer. There's a gentleman who is sunbathing. In this instance, we know exactly the mutation on a DNA sequence level in the RAS protein. It's a CAA CG going to an AT. But here's the interesting thing. It's right next to the thymine, the two thymines. And you'll recall, what's the thing that causes thymines to do something different? Two ones beside. If you were paying close attention, I'll remind you what it was. It's ultraviolet light from the sun. So ultraviolet light from the sun causes these two thymines to bind together. Then all havoc breaks loose in the neighborhood around this. And you get a CG pair to an AT pair changing. When that happens, you get a RAS protein that no longer requires growth factor binding. It's always on, and you can get melanoma. So this is a great summary that, by the way, I just described of the course so far, right? Epidemiology, people exposed to sunlight. DNA damage, thymine, dimer, and errors in DNA, to actual tumor. So in these two lectures, I've tried to describe the overstimulation of normal cells resulting in cancer by oncogenes. I described how viruses can bring oncogenes into cells, and that discovery led to the discovery of similar genes inside of our cells already, the proto-oncogenes, and how in 90% of tumors, these proto-oncogenes become activated to become things that can cause cancer and stimulate cell division. They are the gas pedals. Now I want to turn to the hereditary cancer, and I want to show how that led to the discovery of the other class of tumor-related genes, namely tumor suppressor genes. Thank you.